All right, today we're going to break down one of the most vital molecules in the human body, hemoglobin. You know, that essential iron-containing protein packed inside your red blood cells? Its main job is to shuttle oxygen from your lungs to, well, everywhere else. So let's just dive right into this incredible molecular machine. Just look at this thing. This beautiful, complex structure you see here, floating among the very red blood cells it fills. That's the absolute cornerstone of our ability to live and breathe. It's way more than just a molecule. Think of it as a highly sophisticated delivery truck for the oxygen that fuels our every move. And its design? That's the secret to how it all works.
So how exactly is this little oxygen delivery truck built? To really get it, we're going to take the hemoglobin molecule apart piece by piece. Once you understand the components, you'll see just how perfectly this masterpiece of molecular engineering is designed for its job.
Okay, this chart is pretty revealing. You can see right away that hemoglobin is almost all protein. That's the globin part, a whopping 96%. But the real magic, the crucial functional part, is that tiny 4% called heme. That's the non-protein group that not only gives blood its red color, but, more importantly, it's where all the action happens.
Let's zoom right in on that heme group. At its heart, you've got this complex ring structure, a protoporphyrin ring. And sitting right smack in the middle is a single ferrous iron ion, Fe2+. This little iron atom is the exact docking station for an oxygen molecule. It's really that simple. No iron, no oxygen transport.
And here's how it all comes together. A complete hemoglobin molecule is what we call a tetramer, which just means it's made of 4 protein subunits. For most adults, that means 2 alpha chains, which you see here in green, and 2 beta chains in blue. Each one of these 4 chains holds on to one heme group. So, do the math, one hemoglobin molecule can carry 4 oxygen molecules all at once. Pretty efficient, right?
Now here's something really cool. Hemoglobin isn't some static, one-size-fits-all molecule. It actually changes its structure to meet our body's changing needs as we grow. Let's take a look at how it adapts all the way from the womb to the outside world. So in adults, almost all of our hemoglobin is hemoglobin A. That's the one with the alpha and beta chains we just saw. But you'll notice there are other minor forms like A2 and F. You might wonder, why even bother with these? Well, those tiny changes in the protein chains, using a delta or a gamma chain instead of a beta, they have huge functional consequences that are absolutely critical at different stages of life.
This whole process is just a marvel of genetic programming. This diagram shows us that the genes coding for these globin chains are all clustered together on chromosomes 11 and 16. During our development, these genes are switched on and off in this incredibly precise sequence. It's this genetic choreography that orchestrates a big switch from making fetal hemoglobin to making adult hemoglobin.
And this timeline is really the key to understanding it. While a baby is developing, hemoglobin F is the main player. And why is that? Well, it has a higher affinity for oxygen, which is a fancy way of saying it's better at grabbing oxygen. This lets the fetus effectively pull oxygen from the mother's blood through the placenta. But after birth, you don't need that advantage anymore, so the body makes the switch to the adult form, hemoglobin A, over the first few months of life. We've already said that the iron atom is right at the heart of what hemoglobin does. So it makes you wonder, where do we get this supercritical element? And how does the body handle it? Let's get into the vital role of iron metabolism.
You know, the body's need for iron can vary a lot depending on who you are. Men need about 1 milligram a day. Women, on the other hand, typically need about double that, mostly due to menstrual losses. And for pregnant women, that number jumps even higher to support both their own body and the growing baby.
And here's a pro tip. Not all iron you eat is created equal. Heme iron, which you get from animal products like red meat, is super easy for our bodies to absorb because it's already in that heme structure we talked about. Now, non-heme iron, from plants like beans and spinach, is a bit trickier for the body to grab onto. It often needs a little help from something like vitamin C to really boost its absorption.
If you ever doubted just how vital iron is for carrying oxygen, this chart says it all. A massive 70% of your body's total iron is actively being used inside your hemoglobin molecules right now. The rest is mostly in storage, as ferritin, or being used in other proteins, with just a tiny fraction actually in transit.
Okay, so we know where the iron is, but how does the whole oxygen binding thing actually work? Well, it all comes down to shape-shifting. When hemoglobin gets to the lungs without any oxygen, we call it deoxyhemoglobin.
In this state, it's in a tense conformation, which makes it just a little bit difficult for that very first oxygen molecule to latch on.
But the moment that first oxygen binds, something amazing happens. The whole molecule shifts into a relaxed state, becoming oxyhemoglobin. This change in shape makes it way easier for the second, third, and fourth oxygen molecules to jump on board. We call this cooperative binding, and it's the secret to hemoglobin's incredible efficiency. And that term, reversibly bound, is also super important. The bond is strong enough to carry oxygen, but weak enough to let it go exactly where it's needed in your tissues.
Just let that number sink in for a second. 280 million. That's how many hemoglobin molecules are crammed into one single red blood cell. Now multiply that by the trillions of red blood cells in your body, and you start to get a sense of the absolutely immense oxygen-carrying power of our blood. It's just staggering.
So far, we've basically been looking at a perfectly running system. But especially for those of you studying health and medical technology, it's just as important to understand what happens when this complex structure breaks down. What happens when the blueprint itself has a flaw?
Two of the best-known examples are sickle cell anemia and thalassemia. With sickle cell, a tiny mutation, just one amino acid, causes hemoglobin to clump into stiff rods when oxygen is low. This deforms the red blood cell into that classic sickle shape. With thalassemia, the problem isn't the quality, it's the quantity. Genetic defects slow down or even stop the production of the globin chains, leading to a major shortage of working hemoglobin.
And it's not just genetic flaws we have to worry about. Hemoglobin can also be attacked from the outside. Carbon monoxide is so dangerous because it's a molecular imposter. It binds to the exact same spot on the iron as oxygen does, but its grip is over 200 times stronger. It basically hijacks the hemoglobin, refuses to let go, and completely blocks it from doing its job of carrying oxygen. So with all this in mind, how do we actually check on the health of this system in a real-world clinic? Well, that's when we turn to the lab for a kind of performance review by measuring the concentration of hemoglobin in the blood.
In the clinic, these are the numbers that really matter. A simple blood test tells us if a person's hemoglobin level, measured in grams per deciliter, falls within the normal range. If the numbers are too high or too low, it's a major red flag for conditions like anemia and other disorders. And it's often the critical first clue in making a diagnosis.
So, we've looked at its structure, its function, and what happens when it fails. Hemoglobin truly is a masterpiece of natural engineering. And that leaves us with a final, really important question to think about. What lessons can we learn from its incredibly elegant and efficient design to help us create the next generation of medical tech, like artificial oxygen carriers or new diagnostic tools?